When we're old, genes get turned on that we don't want on, like pro-inflammatory genes, or are the genes that protect us from cancer. Tumor suppressor genes are turned on. And then we see, smart genes like anti-inflammatory or detox genes get turned off.
Right.
We want to be able to sort of stay healthy for the longest haul possible.
Dr. Kara Fitzgerald, I am thrilled that you are on the Better Show. Welcome.
Yeah, thanks for having me. It is great to be with you today, Stephanie.
We are going to have such a productive and exciting conversation. We're going to be talking about your new book, Younger You, and of course, the landmark study that is contained within that book, which shows how we can reduce our biological age with some lifestyle interventions, not the chronological age, but the biological age with some lifestyle interventions, and we can actually age backwards. Before we get to that, I do think, because this is going to be a very meaty, very juicy discussion about aging. And my audience, we were just talking in the pre-chat about how, the type of woman that listens to this show, it's basically like the title of the show suggests, she's looking for information and tips and tricks and strategies on how to get better every day. And we're going to talk about this from a genetic and epigenetic and methylation lens, which can be heavy on the science. So I think it warrants a mini trip back in time to biology, to high school bio. And maybe we can start with just some basic definitions. What is genetics? What is epigenetics? I think that these terms, and I think methylation maybe as well, we require some definition here before we kind of wade into the waters of this.
Absolutely. And if I go down in the rabbit hole too far, feel free to interrupt me, do whatever you need to do to just, get me on straight talk. So our genome is it's our DNA. It contains the blueprint in every single cell inside the nucleus. Rests our DNA, which has it's the blueprint for us. It's just mind-boggling if you think of it. Every single cell in our body has this blueprint. Actually, except for red blood cells, which kick the nucleus out. But it's this extraordinary phenomena. So there is our DNA. And I want to say that in early 2000s, because this will leapfrog into the definition of epigenetics pretty nicely, we finally mapped out all of the genes. We have about 23,000, and that nut was finally cracked in 2002, 2003. 23,000 genes we identified. Now, we really anticipated that this would be the Rosetta Stone for chronic disease. In other words, we would identify, each gene that causes, each condition. So a gene or a couple of genes that cause heart disease, a gene or a couple of genes that cause diabetes, dementia, cancer, et cetera, et cetera. We thought this would be the Rosetta Stone, our answer. And it wasn't. Probably it was a difficult pill to swallow for some scientists, but what it did was usher us into the era of epigenetics. So that is epi, above, genetics, the genes. So this is what regulates our genes, what turns our genes on and what turns our genes off. And this whole entire field, the study of what turns genes on and off, is the field of epigenetics. And it turns out, so a lot of us were of course we were indoctrinated into the idea that our genes are our destiny. The genetic package we were handed from mom and dad, grandpa and grandma, is how we're going to end up. So we're going to live a beautiful long life, or we're going to have, diseases X, Y, and Z. It's written in our genes. There's nothing we can do about it. Well, let me tell you, the extraordinary revolution and the promise of epigenetics is no. In fact, we're sitting in the driver's seat, because what our lifestyle choices, there's a little bit of a heritability component that we can talk about later, but by and large, it's our daily choices, what we're putting in our mouths, how we're living our lives, how connected we are, if we're moving our bodies, are we sleeping, et cetera, et cetera. These are the pieces that influence epigenetics. They influence what genes are on, what genes are off, and therefore influence whether we get these diseases. So the huge transition that we're in the middle of right now is taking ownership and really starting to understand epigenetics and study it. And it will be common language for us in the next few years. Everybody will likely be getting epigenetic studies in their, in their annual physical and so forth. But let me just stop there and see if that explains it.
Yeah, it's beautiful. And I think it's a very empowering message in the same way that, it's not our fault that maybe we inherited poor genes, let's say, but it is our responsibility to maximize their function. Just in terms of history, and just for a complete picture here, who coined and how did this term come about? Epigenetics. Who coined the term, and why is it used so widely today?
Yeah, and so this goes back to, the fact that the same DNA exists in every single cell in the body, but, an eye cell, a retinal cell is very different from a skin cell. So how is this? How is it that the phenotype is so different, even though the DNA is the same everywhere? So it was Waddington, a scientist out of the UK in the 1940s, I think it was in the UK, coined the term because the great epigenetic puzzler was, how does a fertilized egg become a human? How does a fertilized egg turn into the massive different phenotype that is us, that are all of our different cell types? How does such phenomenal change happen from, this little microscopic fertilized egg? And again, all the DNA is the same, but how we regulate what's turned on and off. Each cell type has a different set of genes on and off, so that we have a liver cell and we have eye cells and, cardiomyocytes and so on, and teeth and hair and nails and on and on and on and on. It's just such an extraordinary phenomenon. All of that is regulated by epigenetics. And primarily, and we'll talk about this a little bit more, an epigenetic mark called DNA methylation. And so that was the original puzzler. He didn't know the answer in the 1940s, but he, got enough to pose the question that it's not the genes that are creating this human. It's actually changes to how the genes are turned on and off.
I love that. So save for the sperm in a man and eggs in a woman, let's say nerve cells in the eye or a hepatocyte, they all a liver cell, they all have the same DNA. And the way, we've had, I had Dr. David Sinclair on the show when, his book Lifespan was released. Mm-hmm. And we were talking about, this idea that if we didn't have cellular differentiation, we'd just be like a giant blob of cells. So like the nerve cell in the eye, let's say, you have, and we'll talk about methylation in a little bit more depth, but it's almost like they get like a membership card. Like, here is your card. You are a card carrying member now of the optic nerve, right? Yeah. And it's as we age, we lose the membership card. We actually become more like that giant blob of cells, less differentiated over time, correct?
Well, it's interesting. David Sinclair would say, yeah, he'd say you're absolutely right. And I would say that yes, that's correct, except I would say that there's more predictability. So he would talk about entropy, like there's just kind of random damage that's happening. Yeah. And I think it'd be interesting to ask. I podcasted with him and I'd like to chat with him again, but when you look at the epigenome, so when you look at, those DNA regulation marks of a young person versus an old person, they're almost equal and opposite. And so there's almost this breakdown pattern seems planned. And there is some suggestion that in fact the aging journey is actually a planned sort of programmed phenomena. We have programmed obsolescence. And I think that there's pretty substantial sort of suggestion that is.
So it's not necessarily just like the hard knocks of life and like, yes. But there is some sort of programmable sequence, just like yes. embryogenesis that you talk yes. about, let's say, yes. or like yes. how we can take a newborn, which has absolutely zero function, to a 2-year-old, which is like running and talking That's and jumping right. and skipping.
That's right. In fact, it was Sinclair on my podcast who said that what we see, the changes that we see in aging are sort of as remarkable and grand as what we see in embryogenesis. And even all the life stages, like, well, maybe not. I think that there are some that are the volumes higher, as you said, and there are some that are a little bit perhaps less active. And maybe that's just my ignorance or the lack of science now. But so embryogenesis is massively epigenetically active, and we can talk about specifics around that. It's extraordinary. Early infancy, just like you said, we are aging at an accelerated rate, and it's what we want. We want our kids to be, they're just busting new neurons out and picking up new abilities. Like, within hours we can see changes. Like watching my daughter, like if she got a little cut on her hand as a baby or something, it's like you can see the skin knitting itself together, before my eyes. And then the next day it's pink and beautiful and gone. I mean, it's just so mind blowing that accelerated aging is what's supposed to be happening. You can see developmental delays in kids, and that's a problem and something that we can talk about. I mean, cuddling has been researched, or lack of cuddling, so not just nutrition, but lack of connection and love will alter epigenetics in early infancy and slow down that accelerated rate of aging that's supposed to be happening and result in developmental delay. But we see it through the lifespan. But, according to Sinclair, and it certainly seems in my read on the literature, that early is very, very active. And then as we get older, is also very active. But when you look at it broadly, what's happening when we're old is like the equal and opposite to what's happening when we're young. So when we're old, genes get turned on that we don't want on, like pro-inflammatory genes, or are the genes that protect us from cancer. Tumor suppressor genes are turned on. And then we see, smart genes like anti-inflammatory or detox genes get turned off. It's like, it's pretty predictable and it's a raw deal. So even if we're a healthy older person, we're still experiencing some of these changes. That's one of the reasons why we looked in our research study in a healthy population. I mean, we want to be able to sort of stay healthy for the longest haul possible. And I think it's an important, I think it's a really essential line of inquiry that we have to be undertaking. How do we really keep a robust health span and, concurrently improve lifespan?
And what's the tipping point? So when we have a newborn, we see this accelerated aging, and that's what you want. You don't want developmental delays. You don't want the child to be behind, let's say, his or her peers. And then on the other end of, like, if we oscillate all the way to the other end of the spectrum, then we sort of see this predictable acceleration again, but in the wrong way. So where is it? A
breakdown.
A breakdown. Yeah. So where is it in the span, or maybe this question hasn't been asked, or maybe you have some thoughts on it. Where is the tipping point where we want to, it's like, where's the sweet spot where we're not accelerating anywhere? We're just kind of coasting is not the right word, but like, there's a medium there.
Well, I think that's a really interesting question. I would say that we want to, we're using the same tools to regulate gene expression, regardless of where we are on the life continuum. And we want to therefore be thinking about eating for our genes. We want to be engaged in gene whispering lifestyle practices and dietary patterns that optimize expression really from preconception on. So I would argue that there's no time we don't want to be thinking about these tools. There's no time that we don't want to be introducing some of these habits and foods and so forth to our kids. But I would say that, it's interesting. So when I wrote the book, I said that probably, we're doing pretty well up until our 40s. That was my that was my conclusion on my read in the literature. But if we really want to be healthy centenarians, if we really want to live, to be 120, I would say that we want to engage in this information as young as possible. And when you look at the literature, so I talk about the overcalyx study or region in the book, and this is the overcalyx cohort is a population that has been tracked very carefully with copious data since the 1800s, and they're a farming community. and they could see, they could track health outcomes with lots of food, when they had a good harvest year, and insufficient food when they had bad harvest years. And they could track health. They just track data so carefully. You can look at health outcomes across the generations, and you can see the influence on gene expression when prepubescent boys. So this is altering spermatogenesis. This is altering information on the sperm. This is altering epigenetics on the DNA of the sperm. In prepubescent boys, in that first overcalyx generation, actually influenced health outcomes generations down. So too much food intake influenced generations down, and this is pre-puberty. And we know
crazy. It
is crazy. But we also know that, and this, I know it's crazy. And yet we also know that it happens. Certainly in early infancy, we can see all sorts of, it's not surprising, adverse childhood events will, predictably alter genetic expression and, increase risks of addiction and depression and mental health, but also inflammation and the chronic diseases of aging. So these early experiences can definitely alter epigenetics, towards the better or not. So conversely, so going back to this overcalyx cohort, so prepubescent boys with too much food, so they had fabulous farming, and I guess maybe they had lots of bread and butter on the table or whatever, just more caloric intake, had poorer health outcomes. But the less caloric intake actually had better. So they weren't starving, because starving will set up a whole nother very negative phenomena. So it was almost like they were forced, modest caloric restriction. So not quite starvation, but less food. And the prepubescent boys who consumed less food actually had better health outcomes, and subsequent generations also had better outcomes too. So that doesn't mean that you necessarily want to put your 11-year-old on a time restricted eating structure.
Do not put your 11-year-old on a TRE. Yes, do not do that.
Right?
Yeah.
But it just shows you the power of nutrition in regulating gene expression throughout the lifespan. Yeah, it's extraordinary.
And in your book, you talk also about the is it called the Dutch Winter? It's when when the Germans blockaded the So
Dutch Hunger Winter, that was during World War II, and they shut off access to this very this very specific region, and they were starving. So they were subsisting on little to no food, and women who were pregnant, not siblings, but women who were actually pregnant, gave birth to babies who had increased risk for cardiovascular disease, diabetes, I think schizophrenia, but like just the chronic disease of aging that we see. So there's this thrifty epigenome type. So they hung on to every calorie. So once they had food availability, their bodies were hardwired epigenetically to hang on to calories, and not just that first generation of offspring. Actually, that would be generation one. So the pregnant moms were generation 0, and so that was generation 1. Subsequent generations, they tracked this pattern, over multiple generations, and it's related to DNA methylation and epigenetics.
Yeah. So to your point, you can have the overconsumption, the mild CR, the mild caloric restriction, and then you can go again to the other end of the spectrum with starvation. And we can see with dysregulated nutrient sensing in the body, how that can lead to epigenetic changes, like the thrifty gene you were talking about in the Dutch famine. Dutch winter.
Let me tell you this. I think a lot of us in the United States, many of us are, relatively recent migrants to the U.S. I mean, I think it was my great grandparents who came from Poland, and I write about this in the book. A lot of us probably have some kind of a thrifty epigenome. I mean, if you look in the U.S., what's the percentage of individuals here that are at least overweight? Like over half I think 60%. How many of us are on the metabolic continuum? It's like a river pushing us down towards diabetes, and heart disease and cancer and dementia. That is the trajectory. Yes, some of that is our sedentary lifestyle. Some of that is the standard American diet, of course. And we probably have some inherited patterns. We can actually look at Native Americans, we can look at African Americans and see, similar disruptions. So like Native Americans were, sequestered into reservations and given like flour, just given, horrible processed food and taken from what they were, what they evolved consuming, and just developed diabetes just, in spades overnight. And so you see this, this phenomena elsewhere. But I think all of us probably have some kind of or many of us, I shouldn't say all of us, but some have some sort of inheritance pattern. And we need to really put our minds to fighting that river. And we can make those changes.
Yeah. It's interesting that you say that, because I have part of my background is Mediterranean. So my father was born in Portugal and migrated to Canada when he was young. And the standards of beauty, because he grew up very, very poor, so we also have like most of my cousins, my grandmother certainly, and my aunts. When they came here, of course, we have this abundance of food. There's no, like they weren't in, sort of mountainous or like, villages where, they didn't have the money to buy, whatever. And they've all become, I would call it morbidly obese. Yeah. And it's interesting. This is kind of a side topic, but it just popped in my head as you were talking. Also, what we consider, or I remember my grandmother when I was younger, she would cry when she would see me because she was like, "You're too skinny." Like she was so afraid. And every European grandmother can, like, if anyone here has like, Italian or Greek or Spanish, like she would like pinch my wrist here. And if she couldn't get anything, she was like, "Oh my God, like you're starving, you're going to die." And so the standards of beauty for a Portuguese woman, and of course we know this sort of translates as well into South America as well, is like a larger woman is typically, maybe she's richer. Like there's like this, like, oh, she has a bigger butt, right. Like she's better because she has food to feed it. Right. So just, the beauty standard, it's also that I think that there's also some interconnectivity there as well, because with at least I can just speak for the, sort of what I've observed in growing up in a blended family, but some Portuguese influence there, is that the bigger your bottom is, right? Like the bigger your booty. It's always been, I mean, now of course we have the Kardashians and stuff, but like before it was, growing up in Canada, that was when I was younger, it was like Kate Moss, she was the reign supreme. But my culture, where we come from, is like bigger is better. It means you have food. It means you're kind of like,
you're going to survive. Basically, survive. I mean, it does. It comes from, there's a rational background to that, even if it's gotten a little out of hand. My family also, coming from Poland, like my grandparents had a delicatessen, and, there was just endless pierogies and just excess amounts of,
or it's just like it's like the prototypical, Italian grandmother that's like, mangia, like have some more, like have another thing, Okay. So we've talked about one hallmark of aging, which is like dysregulated nutrients, right? Input. Can we talk about some of the other hallmarks that we see in aging? I know that there's like genomic instability, which we're going to talk about. There's telomeres. Do you want to touch on some of the other hallmarks of aging, and then we can kind of move into the deep epigenome?
Well, so what I would say is, there's a handful of hallmarks of aging. I have focused most of my attention on epigenetics. So we know that there's a protein misfolding, and of course genomic instability, cellular senescence. What else is there? Oxidant stress, inflammation.
Right.
I haven't studied those as carefully as I have epigenetics. And the reason is, so epigenetic disruption, epigenetic changes are one of the hallmarks of aging. But I'm convinced, as I said earlier, I alluded to earlier when I said aging appears to be a program phenomena. If we're looking at root cause, I think that of the hallmarks, probably epigenetics is root cause, or it's the best thing we have to point to root cause. And the rest of the changes are downstream of the epigenetics. So the epigenetics, which is driving gene expression,
Causes
drives these downstream changes.
Right.
Yeah.
So if we have this programmable, in the same way we were talking about this embryogenesis, programmable development of the embryo leading to the, the baby. Yep. And then now, based on what you're saying, we have this programmable aging. What is happening to the epigenome? So let's talk a little bit, first, maybe a definition of methylation. And I'd like to maybe wade into the sirtuin waters as well, but let's talk, if you want to go there. But I think let's talk about methylation, and let's talk about how we start to, what are some of the maybe drivers of this epigenetic degradation?
Well, so, you could, so if you want to think about it from sort of the standard, I think the standard current scientific thinking is that these changes are wrought by, the slings and arrows of life. So inflammation is turned on. Actually, there's another idea that, that genes that we need on, that promote growth and vitality and the good things of youth.
Like mTOR?
Yeah. Mm-hmm. Yeah. mTOR. Yeah. So those things that drive development in our youth, we peak out, you can sort of see a bell curve. They drive forward for a while and they're beneficial. Mm-hmm. And then they actually become damaging to us. And so, blood sugar control and all of the, increased risk of cancer and cardiovascular disease, diabetes, et cetera. All of these phenomena, these pro-growth changes become detrimental and drive the aging process. So that's another school of thought around aging. Actually, it's a pretty well accepted school. And then there's, so there's the influence of just the slings and arrows of life, toxin exposure, sedentary lifestyle, poor food inputs, et cetera. Plus this, the favorable genes sort of turning on us. And then there's those two schools of thoughts. And then there's the school of thought that I'm really kind of encamped with, that this phenomena is predictable and as predictable as, the changes that happen in embryogenesis and early infancy. And that is that there's some sort of drive towards planned obsolescence where predictable genes are turned off, like beneficial genes are turned off, toxic genes are allowed to be turned on. And, this causes us to sort of collectively break down by, somewhere between our 70s and, maybe 120 max. So everybody's still teasing it out. I think, I think we're still teasing it out. Like all of the answers aren't there yet.
I think this is such an interesting conversation because it really has the possibility to like flare up the ego, right? Because it's like, I want to live forever. Like what person doesn't want to live forever? And then we sort of look at some of the conversations around aging in general. And I was saying this to you in the pre-chat. I think that we're really scared of death. As a society, we ship off our elderly to homes because we don't want to see the changes. Funerals are, they are what they are. Like people have a really hard time dealing with death. And I think that, this kind of comes up as well with like consuming meats as well. Like people don't want animals to die and it can get very political and very inflammatory very quickly. But life, a part of life is death. That is really, and I was saying to you, like, and I was saying like the type of person that listens to my podcast typically is like a 35 to kind of 55-year-old woman, where we start to notice that things in our 20s don't work exactly the same way anymore when we are in our 40s or in our 50s and beyond. And I think that there's a lot of resistance to that. Like, no, I want to be like, and I am a vain woman. Like I want to know how I'm going to minimize sirtuin confusion with keeping my skin, let's say, as plump as I can. I want the maximum amount of collagen and elastin. If we're just using like, face, let's say, or skin, like I want the maximum amount that I can achieve naturally. Right. And I think that we want to preserve that, but there's this, there's a beauty in, one of the things I say about, I talk a lot about menstruation and like how menopause is almost like this energetic portal for us to actually say, like, this is who I am now. I'm not like a servant to my children anymore, or to my husband, or to my career. And this is like, I'm going to live my life for me. And I think that this is almost like the second spring, if you will, of our lives. But I think that there needs to be some reclamation of and acceptance of death, that we're not going to live until 140. And even if that were possible, is that really good for the planet at large? For like, I don't know how many people are on the planet now, 6 billion, 7 billion, 9 billion, something like that. Do you want 7 billion, 6 billion, 140-year-olds, and then the next population? Like it becomes this ethical, like, then what, we're already like, people are already saying that there's too many people on the planet. Not that I agree with that, but yeah.
Right. Well, I mean, you said a lot there.
Yes, I did.
So you said a lot. So scientists, some reputable scientists are definitely thinking we're, we could be nearing what they call escape velocity, and that's immortality, which is pretty crazy.
But should we?
So, well, I want to say that, not to overquote David Sinclair, but he said something interesting about, if we can reverse biological age by a year every year, and then we're moving into this escape velocity space. Because we're starting to crack, and I honestly think it has to do with aging being a program phenomena. We're able to, and it also being sourced to the epigenome, we are able to do some pretty radical age reversal in animal models and in vitro models. So, for instance, they caused aging in mice by scrambling the epigenome, and then using Yamanaka factors, which are transcription factors that very specifically influence DNA methylation and demethylation. So they're influencing epigenetic expression and activity, reversed that pro-aging phenomena. So they basically made mice get old, and then they turned them around and made them get young by shifting what was happening in the epigenome. And what they found, these are postdocs out of Sinclair's lab, is that the epigenome retains a youthful record. So it's there. It's like we have our youthful epigenetic pattern with us all the time.
No matter your age, no matter your chronological age.
That's what they demonstrated. So they showed that. They did an animal study where they caused in mice, they caused a age-related optic neuropathy and then reversed it. So that study was out in maybe early 2021. It was published. I think it was in preprint maybe in 2020. But so they caused aging in eyes, and then they reversed that aging. So they made the neurons younger. They also restored sight. And then there's another study that was recently published looking at skin cells, human skin cells in vitro. So they took skin cells. They were from a 53-year-old woman, I want to say, and turned them and reversed them in vitro by 30 years. So we're messing around with immortality big time. And I want to say there's kind of one of the eccentric scientists of the biogerontology space, of the longevity space, Aubrey de Grey, often talks about the, what does he say? I forget his quote directly, but it's like we're brainwashed to believe death. We're brainwashed to believe that age and death are inevitable. Sort of like the opium of the masses or something, that, of course we're going to get old and die. And so I think kind of the radical side of the longevity space believes that immortality is possible and relatively soon. Certainly living healthy into supercentenarian status. Even more conservative biogerontologists are on that. So and let me just tell you this, Altos Lab Altos lab in San Diego. It's also, I think there's a lab set up in Cambridge in the UK and over in Asia. Billions in funding. I mean, it's thought that this is Jeff Bezos and Peter Thiel, like these massive, massive billionaire guys are investing. And this is all about longevity. And they've got the best scientists. So Yamanaka, I just mentioned Yamanaka, he's going to Altos. Morgan Levine, who came from Steve Horvath's lab, including Steve Horvath. So like the top scientists in the biogerontology space, those two are both around developing biological age clocks. They're all going to Altos. Saudi Arabia just committed $1 billion a year to studying longevity. So it's a huge deal. And there's some aggressive, rapamycin, which is when used in high doses is immunosuppressive, but in low doses, it appears to, in, many different animal models, extend life. And I mean, so we're just hot on the trail of, I think changing the life experience here in a big, big way. And we need to be ready for that because it's happening. I mean, before we had airplanes, right? Who could imagine? Even before we had internet, I remember logging on early and what an extraordinary,
Or when BlackBerry was like, you need a keyboard on your phone. And it's like,
so we're at an extremely accelerated, trajectory here. And I want to say some of the more eccentric people, there's this guy, Harold, he's a scientist, Harold Katcher, who worked with Steve Horvath, and he developed this, what he calls Harold's elixir, or E5, and it's components from serum. So they're different, plasma proteins that he's shown to extend life pretty extraordinarily, remarkably in an animal model. And, biological age clocks corroborate that, as do other tests like strength and, antioxidant status and, inflammatory markers and so on and so forth. But he wrote a book recently, and the first half of the book he's talking about this like immortality and how we need to colonize Mars. So to answer your point, what we're going to do with these extra, 10 billion humans, because nobody dies anymore. It's so great. It's so crazy to think about. We're all getting on SpaceX. Get ready. So that's his solution. We're living in some pretty crazy times. I mean, think about CRISPR and gene editing software.
Right.
God, I was reading from a laboratory that produces some kind of biological age clock. I don't know how reliable or reputable they are, but they're in there, and I get their newsletter. Writing about using CRISPR to edit genes so that the sports playing field is level. So if my kid wants to be, a top of her game, soccer star, she could edit her genes to make that possible. I mean, I just find those thoughts crazy. And just to your point, to what you alluded to, it's like fraught with ethical considerations. It's just so fraught with what are the ethical considerations? And to your other point, so that was one huge point, what are we going to do with this? I mean, I don't know. It's just so scary and so grandiose to, try to create superhumans and, think that that's somehow fair and reasonable. I don't even have a place to put that, when I read that in that e-newsletter. God. But then the other point is too, like so we have a youthful epigenetic pattern. And if we can restore that by, taking some kind of a Yamanaka cocktail, and incidentally, originally they would see cancer. So Yamanaka factors, there's 4 of them, and he was able to take a somatic style cell, so a fully evolved cell, and turn it back into a pluripotent stem cell. So it's called an inducible pluripotent stem cell. So he could just turn back the hands of times using this Yamanaka cocktail back to stem cell status. But now other labs, including Sinclair's, are tweaking variations. So they're not using all of them and not turning back the hands of time, to stem cell status, to the blob, as you were talking about earlier, but just taking it back a little ways. Previous experiments resulted in cancer and resulted in, all sorts of like crazy mischief going on as far as, teratomas and, just weird cellular collections. And, it's definitely not ready for humans. But they're certainly working on tweaking it. So your question earlier was like, if we're messing around with this, if we're messing around with our epigenome and our genome, the hard won maturation. So as our bodies break down, right? We, like I've got gray hair, I've got more wrinkles, I'm doing great in some ways. And I can see that I'm 55 in other ways. There's no doubt about it. But what about the maturation that I've gone through? My years living in a Zen center, my movement to, peace with my family, or, just the connectedness that I'm able to experience, my calm, the calm that you get as you grow up a little, is that, though I want to say that we know psychological experience is biologically embedded on the epigenome. That's what it's called. It's called biological embedding. Most of the science around that, like most of the research, is on trauma. There's sort of an obsession with studying the patterns of trauma.
Right.
How do we study the patterns of wisdom? And maturation and connection and love. I mean, there's some suggestion towards that, but we need to be looking there. And to your point, if we revert ourselves back to 20, am I going to be 20 again emotionally, which really wouldn't necessarily be beneficial to me.
God damn it. I was such an idiot when I was 20. Yeah, I don't want to go back there. Yeah.
I heard that this is actually being studied. I heard that this is actually being studied in an animal model. If you, turn back the hands of time, do you lose some of that maturation? I don't know. I mean, and that's a very different model than humans, but so there's much, you're right. Who the heck wants to go back to those years? It's nice to be out.
I would never go back to my 20s. Like never go back. I'm so happy in terms of my life where I am now. And as you were saying, the experience, like that's, and I don't have an official opinion about it yet, but when I hear about like the Bezoses and the Saudi Arabian princes and whatever, it's like, is this just like real life Thanos from Endgame? Like this like big villain who wants all the, if you're not a Marvel fan, I'm not making, my joke is just falling totally flat right now, but it's like, he gets like this, like a stone for like time and wisdom. And like, he becomes like immortal in all these different ways. And it just seems like, I don't know if this is exactly where I'm landing yet, but it seems really immature. It seems really egoic, and it seems really like I want all power always. I never want anything else to come after me that's more important than me. Versus I'm going to do the best with the time that I have. So lifespan aside, I want to have the best health span that I can. I've talked about being like, I want to be the favorite grandmother. I want to be the grandmother that can play with my grandkids, get on the floor, lift them up. If my sons or their, wives or partners, whatever, if they have to work, then I'm like, no problem. I got it. Like I'm strong. I can take them to the park, And in order to do that, I need to be, in order to be a strong, kick-ass, let's say, if I want to be like a kick-ass 85-year-old, I can't be an average 45-year-old, right? So this is where we kind of merge this conversation into nutrition. I would like to anyway, and we can play around with the Horvath clock. We didn't talk about that yet, but we'll circle around it as well. But nutrition and exercise, because those are, if there's ways that we want to even just slow down, let's just assume that we want to slow it down. We know that the Horvath clock is sensitive to things like caloric restriction, which we've talked about, but also foods that we eat, right? Yeah, and 100%. Exercise. So talk a little bit about,
let me, so go ahead, finish your question. I'm sorry.
No, I was going to say, let's talk about nutrition and exercise. It wasn't,
So I want to say, just to tie this back to what we've just been talking about, the Yamanaka factors, et cetera, and really change, and the fact that aging root cause appears to be housed in the epigenome, and specifically perhaps DNA methylation and demethylation. So you were talking about these really kind of crazy things happening in the world now and visions for the future. But one of the extraordinary things is, as you're suggesting, diet and lifestyle heavily influences this same epigenome, these same DNA methylation and demethylation patterns. So what we're doing every day has an influence here. And that's what our study showed. So we looked at we looked at epigenetics and specifically DNA methylation using our diet and lifestyle intervention, and we saw that using very simple, very safe intervention, we were able to reverse biological age. So we slowed down the aging trajectory in our population by over 3 years and 8 weeks as compared to the control group, which is absolutely
3 years.
remarkable.
3 years. I just want to highlight that. Just through food, through nutrition.
Just through food. Well, food and lifestyle. We wanted them to exercise, get enough sleep, and we wanted them to engage in meditation. So the take home of what we demonstrated is Yamanaka factors are not, rapamycin are not, moving to outer space are not, you need to be thinking about these things in our daily life. So there's much that we can do right now and that we should absolutely be doing right now, while all the hubbub happens over there and we can talk about that or listen to, read about it, et cetera. But we need to be doing it. And we know that we absolutely can influence profoundly epigenetic expression. And as we talked about with the Overcalyx study and Dutch hunger winter and so forth, we know that this can influence generationally. So we did, amazingly, we were thinking about epigenetics and DNA methylation in relation to cancer. It was somewhat serendipitous how we actually got to this, because at the time of our study design in late 2017, early 2018, and actually starting our study, we ran our study 2018 through 2019, nobody demonstrated, there was no evidence for biological age reversal in humans at that time at all. We knew that there was a biological age clock available so that we could look at whether we changed things, but I wasn't holding my breath. I was interested in, so my original investigation in in DNA methylation was with regard to cancer, because cancer, really all the chronic diseases of aging, but especially cancer, well, at least there's a ton of research in cancer, hijacks epigenetic expression, hijacks it, takes it over from us, kicks us out of the driver's seat, turns genes on and off right in the tumor microenvironment for its own proliferation. And as a functional medicine clinician, my question was, could there be anything in functional medicine that we're doing wrong? Could we be supplying supplements, et cetera? Could we be sort of pushing this forward? And my conclusion was the safest thing to do was a diet and lifestyle intervention, leaning on that primarily. And then if somebody needs supplements, layering in that, layering those in afterwards.
But the foundational basics are mastered.
Yes.
Yeah. Mm-hmm.
And so what ended up, we were given an unrestricted grant to study this from Metagenics. And what I ended up learning over this journey was that, as I said before, the changes in aging that happen are the changes that happen in cancer, are the changes that happen in heart disease.
Yes.
So we, by entering into the conversation over here, by considering cancer as our original question, we were in fact looking at the changes that happen in aging. And so then our first question was, are we actually changing aging? We will come back and look to see if we've changed some of those genes and that are changed in cancer. And the answer, I can say the preliminary answer is yes. It looks like we've turned back on protective, anti-cancer genes and so forth. And I have to write that up and publish on it. But our first question was, if we're changing aging, if we're making people biologically younger, we should, by extension, be improving health span, improving lifespan, and reducing risk of all the chronic diseases of aging, because aging looks like those. Does that make sense?
Yes, 100%. Yeah.
So we happen to be we happen to be preceding the wave, almost just by serendipity in how we, and then the first study. So we were wrapping up our study, and then the first study that showed biological age reversal in humans was published with huge fanfare. Just as we were, unpacking our data findings. And that was just a group of 9 men. They had no control group. And they did a year-long intervention of growth hormone, metformin, DHEA, I think vitamin D and zinc also. So growth hormone injections, like a long and fairly aggressive protocol. But they reversed biological age by, I think, 2.5 years in that year-long intervention.
It'll be interesting to compare. I would be really interested to compare, for example, we know that for men, we can reach sort of peak growth hormone after about, call it an hour or so of exercise. Like you can really drive up growth hormone through resistance training, for example. It'll be interesting to compare, to have maybe there's a second or like there's a control, and then we have the growth hormone intervention, and maybe there's a tertiary group where it's like, can we just get them to lift some weights? And see what, and I say that sort of tongue in cheek because yes, I'm all for medication when it's required, but it's in and I would think that you share this, you may or may not, but I think that it's then and only then after you've exhausted the mastery of, let's say, eating foods that are going to be really great methyl donors, it's going to be resistance training. It's going to be stress management. And for all the women that are listening here, this is like stress management. Like I'm all for, I lift weights. I will lift weights until my last day. But if you don't take counsel on stress management, because it's silent in the same way that cancer is silent, in the same way that epigenetic changes are silent, you will age faster. And
no doubt about it. 100%. Yeah, absolutely. So I want to just go back to the TRIM study. It was called the growth hormone injection study, and say that his original interest was improving thymus gland. So he's like all about the immune system. And then they layered on looking at biological aging, and he found his finding. And they're studying it. So stay tuned. There'll be more data on that whole protocol.
Super interesting.
But 100%. So we included, so when we designed our program, the diet is, and we'll talk about this in a minute, packed with what we call epinutrients. These are nutrients with evidence behind them for favorably influencing epigenetic expression. Packed. So really every forkful should be speaking to optimal gene expression. But we had a meditation. So we wanted people to practice the relaxation response, just a basic breathing exercise for 10 minutes twice per day. And to your point, the clock that we used in our study, 25% of it is regulated by glucocorticoids. They're called glucocorticoid response elements, and they will influence genetic expression through cortisol. So to me, that suggests that 25% of this particular clock we used is about the stress experience. That's more than any other variable that we studied. So to me, stress is gasoline on the fire of aging, no doubt about it. And we can see the diseases of the diseases of aging, the chronic diseases of aging. We can see, psychic experience, et cetera. We can see that stress just aggravates and pushes forward all of those. So it's potently pro-aging. Conversely, when we bring it down a notch, yoga, meditation, tai chi, et cetera, these are all anti-aging. These promote favorable gene expression. People who are practice meditators are biologically younger. But for those of us who aren't going to retire to the monastery on the mountain, what's really cool and heartening is that just one meditation experience, and we know if you're a new meditator, you've got this crazy monkey mind, but even just starting the experience can change gene expression towards the better. However, if you keep doing it, it's just going to move, like a wildfire through the rest of your genes and influence genetic expression, body wide. So it's an extraordinary phenomena, and we'll get the lasting influence if we continue to get engaged in the practice. So one meditative event can have favorable expression, but we can really turn biological age around if we commit to it. It's extraordinary, and it's very cool. And the same thing with exercise. And let me tell you it's same thing. So one exercise event can have favorable changes. Practiced exercisers are biologically younger. There is some, like if you want to go really over the top, there's some evidence that excess exercise, and this makes sense, can be pro-aging.
For sure.
Right?
Yeah.
We all know. Like I used to be a competitive cyclist, and I would get sick at the end of every season. I always had sinusitis or something like that because I just burned the candle, pushed it for really hard, and my immune system was suppressed after that, and I would get sick. But in general, exercising is one of the most potent anti-aging elixirs that we have. And what's so badass to me is that we can, if we're preconception, we can hand some of that information down to our offspring. Isn't that cool?
I love that.
You can hand that 6-pack ab down to your kid, but you can hand down some of that information. And conversely, if you're not engaging in it, that information is going to go too.
Right. Yeah. So it sort of highlights the saying, you got it from your mama. Like it gives you more bragging rights. Like you got that muscle from your mama. Let's talk briefly about methylation and the Horvath clock. I know we've been talking about repairing the epigenome and nutrient, and then I want to get into like the nutrient rich foods that you looked at, and then
we have to talk about that.
And what is happening. I mean, methylation is sort of this like sort of weird term, I think, to people. It's like, what's happening? There's a methyl group, but it's involved in so many things. So describe what methylation is and how that contributes to cellular differentiation.
So a methyl group is a carbon and 3 hydrogens. Go back to basic chemistry and just visualize a carbon and 3 little hydrogen groups on it. We make this, so we make the compound that houses that methyl group in something called the methylation cycle. And we're familiar with the methylation cycle, even if we're not, because we know B12 and folate are good for us, right? And so that's feeding that methylation cycle, which produces a compound called S-adenosylmethionine. This compound, SAM for short, has a carbon and 3 hydrogens on it. And then SAM is used in methyltransferase reactions. And there are hundreds of methyltransferase enzymes in the body. We do all sorts of, so just think that methylation is happening, in the background in really every cell of the body all of the time. It's very fundamental. It helps us clear out and metabolize drugs, clear out toxins, metabolize hormones, make neurotransmitters, just all sorts of activity is reliant on these methyltransferase enzymes and this S-adenosylmethionine. So this is essential foundational work. When we look at epigenetics, we can see that there's a DNA methyltransferase, and there's also methyltransferases in mitochondrial DNA. And so we're using methylation in gene regulation all the time. There's interestingly a set of enzymes called 1011 translocation enzymes that demethylate. So my area has been focused. My attention has been on DNA methylation, the family of DNA methyltransferases, as well as these 1011 translocation enzymes. So we want to have the methylation cycle whirring. We need that functioning. And as we age, it's less efficient. So we want to be eating a methyl donor rich diet. That's essential. If you look at the nutrient breakdown of the Younger You program, you'll see that it's packed with sources of folate, sources of B12, other nutrients that work in the methylation cycle include choline, betaine, even DHA from fish can help regulate methylation cycle. And there's all sorts of minerals like zinc and magnesium and potassium that are needed for a functioning methylation cycle. So fundamental to healthy gene expression, healthy DNA methylation is a nice functional methylation cycle. But interesting is, how is methylation happening at the level of the DNA? Like if we just push methylation forward, if we just take in a whole bunch of folate and B12, et cetera, are we directing where the methylation is happening? And that is where it gets interesting to me. So methylation decreases as we age. And one could say, well, let me hop on a good B complex. Let me take extra B12. Let me take extra folate, et cetera. And a lot of us have done that. But the outcome there hasn't been uniformly beneficial. There's some suggestion in the literature that disordered methylation could happen. So as we age, there's less methylation. But as I've been saying, when you drill down and look at DNA methylation, you can see there's extra methylation happening in some genes and less happening on others. So there's disordered methylation. There's hypermethylation of, again, our tumor suppressor genes, the genes that protect us from cancer. So if we're just taking B12 and folate, could we inhibit our tumor suppressor genes and then actually drive cancer forward? And I talk about this in the book quite a bit. And there's some suggestion that, in fact, this could be true. So we don't want to just willy-nilly push methylation forward. We need to do it with some sophistication. Our discovery, our sort of big aha discovery, was that there are a ton of nutrients, also epinutrients that we call methylation adaptogens. Actually, a friend of mine, Michael Stone, coined this term. We're both on faculty at IFM, and I was talking to him about this discovery, and he's like, "Wow, it sounds like methylation adaptogens." So these are compounds. These are mostly polyphenol compounds that seem to direct where methylation is happening on the genome towards a more favorable profile. These are compounds that you're already eating. All of us are eating some of these methylation adaptogens, curcumin in turmeric, or EGCG in green tea, luteolin, lycopene in tomatoes, quercetin, what else? Resveratrol. So going back to some of these guys being sirtuin activators or supports. Sulforaphane in cruciferous veggies, diindolylmethane, again, also in cruciferous veggies, and on and on and on and on. There are so many of these polyphenol compounds that are epinutrients of extraordinary proportions. So they have nothing to do with the methylation cycle, but what they do is appear to influence the DNA methylation transferase enzymes, in some cases inhibiting them from shutting off a gene, or supporting a gene being turned back on. And it's amazing. So in the literature, mostly the research is in animals and in vitro, but we see, so going back to these tumor suppressor genes that get shut down as we age, we see that these beautiful compounds, curcumin, resveratrol, EGCG, turn on previously suppressed tumor suppressor genes. So they protect us from cancer right here at the level of DNA methylation in the methylome. It's extraordinary. And I want to say that actually exercise does the same thing. Exercise is like a vegetable. For those of you who are
like a
resistant
green leafy
to
vegetable with a side of blueberries.
Is.
Yeah.
Yeah, it really is. It has the power to influence these turn back on gene expression, favorable gene expression patterns. And I'm sure that that's one of the reasons it's an anti-aging elixir. My suspicion with these beautiful polyphenols that we know are anti-cancer, are anti-inflammatory, are antioxidant, or et cetera, probably the mechanism exerting these downstream effects has to do with influencing gene expression. And one final thing I want to say is, in our study, what we saw in our participants was that we saw this hypothesis that we're throwing out. We didn't increase methylation on the methylome. So we didn't, when we looked at almost 1 million methylation sites on the genes, and we didn't net increase it, but rather we moved it around to a more healthy and youthful pattern using a methyl donor rich diet plus these adaptogens and then the lifestyle pieces.
So you mentioned sulforaphanes, you mentioned EGCG in green tea. I'm assuming berries are part of this polyphenol group as well.
Yeah. There, in fact, in the book, there's a nutrient appendix that is the first of its kind, I'm sure, that's full of all of the epinutrients. It's 30 pages long. And for people who are a little skittish about starting a diet, I tell them, look, get the book or borrow somebody else's, go to the nutrient appendix, and you'll see that you're already eating, dozens of these foods, and you can turn the volume up on them a little bit and continue to. And then, when you're ready, you can dive into the full program. But yeah, most recognized polyphenols are gene whisperers. They're epinutrients.
That's great. And I think it sort of lends as well, because you see a lot of times, at least now anyway, I see this and I get asked this a lot because I have a diet which is very green rich, like very sulforaphane and dim, as you're mentioning, like a lot of green leafy vegetables, lots of peppers, lots of things like lycopene and the tomatoes and such. But you do see now that you should avoid plants because they're toxins. You do see like the oxalate, production.
Yeah, I think it's a mistake. I got to be honest with you.
I agree.
I think that's absolutely,
yeah, I agree.
It's just, and we have blue zone data. So we already know we can go to populations where, there's a high percentage living, these beautiful, robust, healthy lives. And you can look at what they're eating, and it flies in the face of some of the fad diets here. which, there's oxalate anxiety. And I think that's just absolutely
Oxalate
overdiagnosed.
anxiety. I love that.
Huge. There's a lot of oxalate anxiety. I mean, we get pinged in the younger you. What about the oxalate?
Yeah. And for some people, like maybe 5 to
a minority.
Like 5 to maybe 10, maybe I'm pushing it at 10% of the population. There is a sensitivity to it. For most people, all you have to do is steam your spinach and like your problem, we have no more problem.
Well, and what's the source of the issue? I mean, it is, we are in a time of really tough guts, and imbalanced microbiomes. And likely that's the foundational driver. And so we should all be able to handle oxalates. Maybe we've got, a genetic predisposition towards stone formation, and we want to be a little bit mindful because of that. But, by and large, most of us should be able to resolve any oxalate sensitivity. And it's misdiagnosed. So somebody has another food sensitivity, or they've got some SIBO, and it gets pinned on oxalates erroneously. The idea that beans, now in our intensive, we don't have legumes on the 8-week intensive, but we definitely want people to migrate into eating legumes and beans and so forth once they finish that 8-week intensive. I don't think we need to be sweating the anti-nutrients if we soak our beans. And again, when we look at those living well and long today in our toxic world, they're eating a boatload of legumes and they're eating high oxalates and so on and so forth. They're actually eating bread and things that a lot of us stay away from.
And I think that there's, I think that there, and I'm just really speaking more about kind of like the carnivore sort of movement here, because I think there's a time and a place for, as you were saying, like if someone has SIBO or they have, hyperintestinal permeability of what am I trying to say? Hyperpermeability of the intestine is what I'm trying to say. I think there can be a therapeutic intervention where we, let's say, remove let's say vegetables that may be causing, that may be causing adding like the gas or the kerosene to the fire, let's say. But I do think that they do have to be in there long term. Like there's too much data, there's too much literature that talks about all cause mortality reduction with consumption of the foods that we're mentioning.
Mm-hmm. Yeah, no doubt about it. And there may be some preliminary cleanup that needs to happen without question.
Yeah. One other thing I wanted to end or kind of start wrapping up our conversation, because one of the things I find so interesting about you in doing this work is that you became a first-time mother when you were 50. Is that correct?
Yeah.
So I feel like this is so interesting. And like, I want to make sure that we touch on this because when you have a young child, when you're, let's say 25 or 30, I was 33 when I had my first baby, there's a certain, like, you just naturally have a little bit more energy, like just because, the methylation is maybe a bit more efficient. We have just more ATP production, et cetera. Mitochondrial dysfunction is maybe not as big as someone who might be older. And I think that this work, I would love for you to speak to your experience as a first-time mother in her 50s, knowing what you know about methylation and how foods can really turn back the clock. And maybe if there's, I'm not sure if it was an adoption or if it was a biological child. I mean, you might not want to speak about either of those, which is fine. But I would just love for you to speak to your experience as a woman in her 50s, being a mom for the first time, and how this information has directed your behavior.
Yeah. Okay. So she's adopted, and what can I say about it? I live what I preach. I mean, I absolutely do. And so my biological age at last check, I get it maybe every 6 months, which one needn't. I'm just kind of particularly interested in it. Is 41. I don't. So I'm chronologically 55, and my current biological age is 41, and hopefully it's going to stick around there. We'll see. I'll test soon. I feel great. So I don't have any limitations with my daughter at all. I think, and I just, and I pay attention. I mean, I just pay attention to the science. I am interested. I need to be a healthy centenarian. I want to be, because I want to be here for my for my kiddo in the best possible way. So all of this whole area is extremely personal and important to me. What else can I say about it? There was another little tidbit I wanted to say. Okay, so going back to our conversation around wisdom, I am different as a 50-year-old, first time mom at 50, and now I'm 55. Yeah, I do have. I approach parenting differently than I would have when I was younger. So I have a nanny. I have more assistance. Like I value. I mean, I see what my mom did, and my sister-in-law, who was the primary. She was the stay-at-home mom, my brother's wife. They were with their kids all the time, and I'm with my daughter a lot. For a business owner and a full-time worker bee that I am, I have a lot of time with my kid, but I also appreciate getting my time into exercise. My self-care is a priority now in my 50s in a way that it wasn't if I had her younger. So that's important. And I also have way more patience now, than I did when I was a kid, when I was younger.
It's that wisdom.
Yeah. I mean, yeah. I would have been a very different parent. I mean, I think I would have been, I would probably be needing to do some cleanup now with my kid based on how I was when I was that age. So there's some benefits, I think, to being an older mom, both for me and how I engage in my own self-care, which I wouldn't have when I was younger. I wasn't aware of it enough, to just, being gentler and just, I don't know, I think just a little, having a little bit more wisdom. Certainly than my younger self. Yeah.
Yeah. And I think that's some of the epigenetic transfer that we don't want to lose. We want to keep that wisdom because your daughter now is benefiting from that wisdom that you've accrued over time. The person that you were, let's say when you were chronologically 41 is different now than the woman that's 55. So I think.
Yeah, 100%. And the other piece that's kind of cool too, because she is adopted, and this one, actually, I was talking to my agent. This was a piece of our book, my book proposal, kind of when it just sort of blew her mind what we were doing. But our epigenome, so I don't share my genome with my adopted daughter, but our epigenomes, your environment converges gene expression. And so we have a shared epigenome. There's just no doubt about it. And when I was telling my agent this story, I had just started reading about this. This is the idea of biological embedding and the environment and interconnection and connectivity and relationship and all of that, how that can influence epigenetic expression. Of course, my diet is similar to hers and so forth. There's other variables in there. But I was telling my agent, and it was a little bit of a risk. I was like, this is a little bit out there, but I'm gonna tell her. And there was silence on the other end of the line. So I was pitching her our proposal for the book, and there was total silence. And I remember thinking, I was embarrassed. I was like, God, I shouldn't have said that. This was just too out there. And she finally said that she was crying. She was in tears because she's got an adopted daughter, and this would be so meaningful, this information. Her adopted daughter is from China, and she's not, my agent isn't Chinese. She's of European descent. And this was just such a beautiful and powerful thing of how they're connected genetically. It's different, but the connection is there nonetheless. And yeah, it was really kind of a beautiful moment for us.
I'm so happy. I just love hearing that because I think that, we often think about, and I'm like kind of getting a little teary right now. Like we often think about motherhood as this very limited, you do it at this time and it looks like this. And I just love everything you're saying. So thank you very much for sharing that and for your work as well. If people want, are you doing ongoing studies right now? Is there another study that you're working on, or this is it for now?
Yeah. So no, people should come to youngeryouprogram.com for the details. And, we are going to be building out groups. There's lots of stuff happening. If for anyone who wants to get involved and do this, if they want to do a real concierge version and have access to the DNA testing we used in our study, et cetera, they can. But we do have approval to continue to study within an app. We've created an app called 3YY, 3 years younger, and you can, your data can be used for us to continue to publish on. And in fact, I will say that we have a little prelim data of a group of 7 middle-aged women who just rocked it. They did very well. So I'll probably publish them as a case series as soon as I get a minute to write it up. But we're just continuing to see really cool stuff.
This has been such a pleasure talking to you, Doc. I'm so happy you came on here. I know this is going to be so useful for so many people listening to this. And I think that we've touched on a lot of, we've kind of talked a lot of mechanisms and mechanistic, but I think there's been a beautiful blend of like philosophical and future pacing and, the kind of back to the original piece that we talked about, like what an empowering message that you can be 3 years younger with how you move and what you eat and some breath work or meditation or some type of stress management. And I think that is so exciting, exciting. And I can't wait to see what else you bring to the table. So Dr. Fitzgerald, thank you so much for your time today. I can't wait.
Thanks for having me. It's just been a pleasure to be with you today, Doc Stephanie.
Okay, I want a Benjamin Button with you. I want to get younger as I get older. Let's figure out how to do it together by clicking the link right here.
Is the Western, now global diet higher in sugar, leading to inflammation through multiple mechanisms, changing our decision-making to being short-sighted and self-centered?